In this video lecture, we will be learning about carbohydrate metabolism disorders. Carbohydrate metabolism is essential for maintaining energy homeostasis in the human body. When this process is disrupted due to genetic enzyme deficiencies, the result can be a group of inherited conditions known as carbohydrate metabolic disorders. These disorders affect the body's ability to break down, store, or utilize carbohydrates, often leading to serious health complications if not managed early and appropriately. One of the most well-known disorders in this group is glycogen storage disease type 1, also called von Gierke disease. It results from a deficiency of the enzyme glucose 6-phosphatase, which is vital for the final step in glycogenolysis and gluconeogenesis. Without this enzyme, the liver cannot release free glucose into the blood, leading to severe hypoglycemia, lactic acidosis, hyperlipidemia, and hepatomegaly. The responsible gene is G6PC. Management involves frequent feeding with slow-release carbohydrates like uncooked cornstarch and strict dietary control to maintain blood glucose levels. Hereditary fructose intolerance is caused by mutations in the ALDOB gene, which encodes aldolase B. A deficiency in this enzyme prevents the proper metabolism of fructose 1-phosphate, which accumulates in the liver and inhibits key metabolic pathways. Symptoms occur after fructose ingestion and include vomiting, lethargy, and liver failure. The only effective treatment is strict lifelong avoidance of fructose, sucrose, and sorbitol. Galactosemia, another condition, is typically caused by mutations in the GALT gene, affecting the enzyme galactose 1-phosphate uridyl transferase. This leads to toxic accumulation of galactose 1-phosphate after ingestion of lactose-containing foods. Affected infants may present with jaundice, hepatomegaly, cataracts, and sepsis. Early diagnosis through newborn screening is critical, and treatment involves immediate and lifelong elimination of dietary galactose, including dairy products. Glucose 6-phosphate dehydrogenase, G6PD, deficiency is an X-linked condition caused by mutations in the G6PD gene. This enzyme is crucial in the pentose phosphate pathway, especially for protecting red blood cells against oxidative damage via NADPH production. Deficiency can lead to episodic hemolytic anemia triggered by infections, certain drugs, or fava beans. Avoidance of known triggers and prompt treatment of infections are the main management strategies. Classic lactose intolerance, while not as severe as other disorders discussed, is also part of this category. It stems from reduced expression of the LCT gene, which encodes lactase. This leads to gastrointestinal symptoms upon lactose ingestion, such as bloating and diarrhea. Management is straightforward, limiting or eliminating lactose-containing foods and using enzyme supplements if needed. Lastly, glycogen storage disease type V, or McArdle disease, results from mutations in the PGM gene encoding muscle glycogen phosphorylase. This enzyme deficiency impairs glycogen breakdown in muscle tissue, causing exercise intolerance, cramps, and myoglobinuria. While there's no cure, patients are advised to avoid strenuous exercise and consume simple sugars before activity to reduce symptoms. These disorders underline the critical role of genetic and enzymatic function in carbohydrate metabolism. Early diagnosis, often through newborn screening and genetic testing, paired with targeted dietary and medical interventions, can prevent serious complications and improve long-term outcomes. Don't forget to like and subscribe to the channel. Dive into our curated playlists and tune into our weekly podcast, where we cover the latest in health, microbiology, and science-backed insights designed to keep you informed and inspired. We will make available practice quiz to test your knowledge. Until next time, keep learning.